Welcome everybody. This is the last piece of day 2, focusing on metabolomics. So we've been going through a lot of concepts, methods, analysis approaches for looking at metabolites, organic matter, thermodynamics. The hands-on session was looking at modeling approaches, which is also Hyun touched on this in his tutorial before the hands-on session. And so now Tim Scheibe, who you all know at this point, I believe, is going to talk to us about going beyond just thinking sort of abstractly about these things and having some toy models to work with and things like that, to really thinking about how can we take these methods to do science, to do numerical experimentation, to learn as we move across sites and to generate transferable understanding in so doing. So with that, Tim, please take it away.
Okay, thank you, James. Great introduction. So hopefully you all can see my screen. Somebody yell at me if not, but this is my title slide. And as James mentioned, what I'm going to try to take is a little bit bigger picture view. So for most of the last 2 days, we've been really down in molecular scales, thinking about genomes and masses of individual molecules and their molecular composition and how that affects microbial reaction rates. And what I'd like to do is sort of frame things up for the next couple days to start thinking about how we might use this information in integrating with reactive transport models to gain understanding about ecosystem functions. And in particular, here we'll talk about in the context of rivers and watersheds. So we'll get started here. Let's see if I can get my slides to work. Okay, so the concept or motivation behind this is really was really brought out when we started getting a lot of traction with WHONDRS. And as WHONDRS expanded and started developing these distributed data sets, one of the things we did early on in WHONDRS was think about how to design the data collection to be useful in models, and particularly in biogeochemical models of hydrologic systems. And so we collected data that was relevant for those kinds of models. And as you've seen over the last day and a half, we have some methodologies to begin to incorporate that information into our models. And so we started thinking about really dreaming beyond just collecting the information and the data, but trying to start thinking about how to use this to develop transferable understanding across a variety of systems. And so what I mean by that is, if you look at the photo here on the left, this is a photo of a field site here in near Pacific Northwest National Laboratory in the Hanford reach of the Columbia River, where over the last few decades, we've done a lot of research on this particular site. We've gained a huge amount of understanding of the hydrobiogeochemistry of the site, how the river interacts with the subsurface environment, how it impacts contaminants that exist in the groundwater, how it affects temperature regimes in the river and in the subsurface, and river water quality. And in the course of doing that, it's been really exciting work, but we've always been thinking in the back of our mind about, well, how can that information be transferred, or how transferable is that knowledge to other sites or to larger scale problems? And so as we've developed a strong research community within the DOE research programs that we work, we've begun really thinking hard about open science and coordinating that in a way that is designed to develop transferable understanding by integrating models and data. And you see a whole number here in the middle of different organizations and resources that have been converging together. You can see there's an Open Science by Design workshop that was done by the National Academy. There's also an Open Watersheds by Design workshop that came out of the Department of Energy that James and others here co-led. And so we've really been thinking about how to incorporate that to now begin to take the understanding that we've learned individually at study of a number of sites and put that together to gain understanding at basin scales, continental scales, global scales. So how can we begin to do that? Well, first we have to think about, well, what are the real critical modeling challenges that we have? And so if we think about watersheds and hydrobiogeochemistry, really hydrobiogeochemistry is the nexus of how hydrologic problems, problems of water flow and transport, impact on biogeochemistry and vice versa of our systems. And so it's really that nexus between hydrology, biogeochemistry that we want to study in our watersheds. And the key question that we want to understand is how can we quantify watershed biogeochemical function? By biogeochemical function, what I mean is really how does a watershed transform either the material, the energy that's brought into that watershed, the water that comes into that watershed and moves through the watershed and ultimately exits the watershed. How does the watershed act to transform those materials, to develop new materials, starting with, for example, the water that comes in the form of precipitation, as well as the energy that comes in and is turned into photosynthetic materials that become organic matter in the system, the elements that perhaps weather from the soil and the rock, all of those things together form the biogeochemical function of a watershed. Now we want to be able to understand that and quantify it in our models, and then having gained that understanding, be able to use that understanding to predict changes in response to future disturbances. And this question is posed or is opposed by a really major obstacle, and that is that there are many uncertainties that are inherent to large-scale watershed models, and these challenge our ability to really understand and predict biogeochemical function of watersheds. And to outline just a few, we might think of them in different sort of groupings. So if we think of the things that are perhaps least uncertain in terms of watershed models, we can measure things fairly accurately like watershed geography. So we have high accuracy digital elevation models of probably the entire world. We have a very high resolution representation of the geometry of river networks, at least in 2 dimensions. We have a pretty good handle on what the land use and land cover of watersheds is through satellite data and other types of remotely sensed data. So these types of data are relatively uncertain. If we go to data that are more moderately uncertain that we need to drive our models, we would think about things like hydrologic fluxes. So in the terms of surface water, these might be less uncertain. We have pretty good river monitoring networks that tell us what the flows are in our rivers. But when it comes to the subsurface environment, it tends to be fairly difficult to interrogate, kind of opaque to observation. And so measuring large-scale groundwater fluxes or groundwater surface water interactions can be quite challenging. Measuring the hydrologic inputs like precipitation and losses like evapotranspiration are also challenging, and especially when we want to predict those into the future. And then biogeochemical inputs such as, for example, non-point sources of nitrate, which is a key contaminant in our surface water systems, can be also moderately uncertain. Now, the things that I think are highly uncertain are things like subsurface residence time distributions. How much time does a parcel of water that enters the subsurface environment, either by infiltration through soil or through movement through a riverbed into an aquifer, how long does that piece of water spend in the subsurface environment? How far does it travel during that time? Where does it emerge back into the surface environment? And along the way, how do the solutes that are carried by those parcels move and get mixed with other solutes and react along the way? But probably of the greatest uncertainty is really the biogeochemical reaction stoichiometry and rates. And so as we think about modeling our systems, one of the things we commonly come up against is how are we going to decide what reactions are important where and what rates those reactions might be occurring. And so really this has been, I would say, the biggest piece of uncertainty. Now, as you think about the things we've been talking about over the last couple of days, this is really where we focused our attention, is on understanding reaction stoichiometry and reaction rates in a way that we can use that in models. And if we can seriously address the spatial and temporal heterogeneity and variability of reactions that occur in natural systems across space and time, that would address probably the largest source of uncertainty in our models. And so this is really what we're aimed at. Now, when I talk about biogeochemical reaction stoichiometry and rates, really we're talking about, first of all, what reactive components exist in our system, and what reactions are acting to transform those components as they interact. And so Hyun's talked quite a bit about this today, about defining the stoichiometry of reactions that impact organic matter, aerobic respiration, and formulating reaction networks out of our knowledge of those systems. And we might represent that reaction network in some sort of pathway representation like a map, or we might represent it as a matrix type of representation where each reaction is represented as a column, and each metabolite that contributes to those reactions is represented in the rows, and the coefficients of those represent the stoichiometric coefficients of our reactions. But in addition to the stoichiometry, we also need to know, in many cases, the rates. And that is, what are the expressions that govern the speed at which these reactions proceed? And again, Hyun has mentioned quite a lot about this today. We've talked a lot about reaction rate models, such as the one shown here, where we can derive these Y parameters directly from a knowledge of the elemental composition of our organic matter. And so as we begin to get a good handle on the stoichiometry and the rates, and we can determine those based on actual observations that we can make in a distributed way, now we can start to get a handle on the distribution of those biogeochemical rates processes across our systems of interest. Of course, if these reactions tend to be very fast, then we don't need to have rate expressions, but most biologically mediated reactions tend to be kinetically controlled. So the question then becomes, can we actually generate local reaction networks and estimate these rates directly from distributed molecular data? And this is really a question that our team has been working hard on. So you've heard from Kelly on day one quite extensively about the metagenomics, from Hyun today about metabolomics and how to incorporate that into models. And Xingyan Chen, our colleague here at PNNL, who you'll hear from on Friday in one of the application presentations, has been working to take the information, working closely with Hyun and Kelly to integrate that information into software that's used by our community within what's called the Ideas Watersheds project. And I'll talk a little bit more about that. So this really kind of starts going back to what Will talked about today, the FTICR mass spectrometer at EMSL that we use to characterize organic matter, as well as other methods such as NMR. And so we can characterize with high resolution the organic matter in our systems, and with relatively high throughput as well, which leads directly to the kinds of models that Hyun has been discussing today based on thermodynamic theory, lambda theory. And as we learned yesterday, we can also characterize the metagenomic potential of those communities that we observe in those samples using tools developed by Kelly's team and by the Joint Genome Institute. And by putting those together into the K-base system, we can, I think we'll learn more about this tomorrow, we can actually build up detailed reaction networks that describe the communities, the metabolic potential of the communities that exist in our systems. And we can do that in a distributed way. Now those K-base tools include metabolic modeling methods that is the subject of tomorrow's presentations, including things like flux balance analysis that we can use to actually tell us what the rates are under given conditions and given particular metagenomic inputs and certain metabolites in our system. And when we have those complex genome scale models, Hyun has been working on developing a way to represent those using a simpler representation, in this case a neural network. And a neural network model can use as inputs the outputs of these flux balance analysis models, and then generate outputs, things like these reaction rates that we need for reactive transport models. And these can in turn then be put into reactive transport models that are being developed under the Ideas Watersheds projects. So again, these links are available in the Google Doc. I'd encourage you to explore some of these resources as you have time and interest. So as we've learned about these things, I'd like to take you on a little bit of a thought experiment using the Columbia River Basin as our example. And this is based on work currently being undertaken by what we call the Subsurface Biogeochemical Research Scientific Focus Area Project, or SBR-SFA. You see the URL there on your screen, and if you'd like to learn more about our project, you can visit that website. What we're doing there is a lot of the kinds of things we've been talking about here, and I'd like to give you an example of how we might apply these ideas. So if we start with, for example, a prediction of stream nitrate levels generated by an existing watershed scale model called Sparrow. This is a model that's widely used. I think it was developed by the US Geological Survey. And Xingyan Chen and her team, including Kyung-Ho Sun, have applied this to the Columbia River Basin. By the way, the Columbia River Basin is shown on the map in the upper right of your screen in the black outline. You can see that this basin covers parts of several Pacific Northwestern parts of the United States, as well as up into the Canadian provinces. And so this part here just shows the United States portion of the basin. This basin is a major river basin within the United States, and I think the Columbia River itself is the fourth largest river in North America. And so this is an important basin in many ways. And so as we are able to make an initial prediction of nitrate in the streams across there, the first thing we might do is apply a WHONDRS approach to gain distributed samples and analysis of those samples across the network in collaboration with our partners to understand metabolomic, metagenomic, and water quality variations across the basin. And this is where the first stage of our so-called MODEX cycle can come in. MODEX is the integration iteratively of models and experiments in a cycle in which they inform one another back and forth. And so here, for example, the initial model might help us to decide where to take our samples. These, by the way, are just randomly placed here. They're not representative of actual locations that we're sampling, but in general, we can use a model to guide the initial placement of our samples and our collections, and then we can use the sample results themselves to test the predictions of our model. In this case, what are the nitrate levels that are distributed across the basin? Now, if we take the metabolite information, the metagenomic information, and put that into KBase, we can begin to generate reaction models that can be solved using tools like pFlowTran to actually estimate local rates and how they might vary as a function of hydrologic variations. And again, we have the second stage of our MODEX cycle where we can again couple that to experiments. So if we've predicted a particular reaction is occurring at a particular rate, we might test that against laboratory experiments using samples from those same locations. And this is an example shown here of our laboratory batch experimental system that allows us to run many experiments at the same time. Each of these vials is an experiment looking at the aerobic respiration in that particular vial. And it's a semi-automated system in that these vials have on the ends of them little optode tabs, and an optode is a material that is sensitive to the oxygen in the inside of the system and is sensitive in a way that changes its color. And so when the camera images these, it can actually determine the oxygen concentration without removing samples from the bottles. And so we can get a semi-continuous measurement of oxygen in a large number of samples at the same time. And so this gives us a way to begin to verify and test some of our numerical models. So then the question becomes, is there a way to take these samples, which even though they are distributed, are still limited in scope, and extend that over the entire area of the basin? And so one of the ideas we're approaching is to think about classifying the basin into different subdomains, different classification types, depending on using machine learning applied to remotely sensed data sets, such as vegetation cover, elevation and slope, different aspects of hydrology, transpiration, and so forth. And by doing that, we can classify into different watershed classes, but then if we've got enough samples in each of these classes, we can hopefully gain some understanding about how the biogeochemistry of these systems varies as a function of class, and use that to extrapolate across the spatial domain. Ultimately, with the goal then of predicting large-scale cumulative effects across the basin of things like total denitrification rates and the production of carbon dioxide. Now, these models are preliminary models that don't yet incorporate any of these distributed metagenomic and metabolomics data, but our objective and approach is to iteratively update these models as we gain new understanding through a WHONDRS style approach coupled to numerical simulations. And once we actually have models that we have tested and gained confidence in through this MODEX cycle, we can begin to use those models to explore what-if questions through numerical experimentation. So a couple of examples. We might ask ourselves, what if the hydrologic regime changes within the basin? It might change due to, for example, climate change effects, which will lead to either hydrologic intensification in some areas of the basin, or could perhaps lead to drought, or maybe could lead to changes in the timing of snowmelt and the amounts of snowmelt relative to rainfall. If we can understand that, we can put that into our model, or we can predict what that might have as an outcome. One particular type of hydrologic disturbance that we have been studying is the effects of hydropeaking by hydroelectric dams. And this particular image shows data that describes hydropeaking in the Hanford Reach of the Columbia River. This is about a 70-kilometer reach downstream of Priest Rapids Dam. Priest Rapids Dam is a hydroelectric dam that generates electricity, and it does so by releasing water through the turbines in response to electric power demands. And therefore, the amount of water released at any given time changes quite dramatically over the course of a day. In fact, the discharge of the river downstream of the dam can vary by easily a factor of 2 over the course of a day. And you can see that also leads to changes in river stage or river water surface elevation, which is what this particular figure represents. This is a time series starting in 2011 through 2015 of the variance in water stage in the river as a function of the vertical axis being distance downstream of the dam in kilometers. And what you can see from this is that the heat map shows that there are periods of time in which that hydropeaking variation is accentuated. And you can also see that that effect is dampened as you go downstream from the dam, as one might expect. So we see here that in November to December timeframes, we tend to have the most effect of the hydropeaking on the river stage variations. That river stage variations leads to changes in inundation along the riverbank, as well as changes in exchange fluxes between the river and the groundwater. You also see an interesting feature over here in 2015, which was a low flow or a drought year. And we see that those hydropeaking features are actually exaggerated and even become bimodal during that drought year. So we can begin to see some of the effects of drought or of different hydrologic regimes on how our biogeochemistry of our hyporheic zone can function. Another really interesting disturbance we might consider with our what-if questions is the effects of wildfires. As many of you may know, wildfires are a very significant problem in the Pacific Northwest and the Columbia River Basin and across the western United States and Canada, up into Alaska. These are seasonal effects that become very strong this time of year, and we often have very severe wildfires burning across the region. This particular wildfire is one of the largest fires in recent history, and it occurred along the Methow River in the upper Columbia River Basin. And wildfires have many effects on hydrobiogeochemistry of river corridors and watersheds. One of those is that as the organic matter is burned, it changes its molecular form and character. And as you've heard a lot today, the molecular form and character of organic matter matters a lot in terms of how it impacts the biogeochemistry of the system. And so one of the things we're studying then is, as we have pyrogenic organic matter coming into our river system, how will that affect the biogeochemistry. If we have a way, a pipeline that we can use by characterizing the organic matter to predict the biogeochemical outcomes, this will help us to ask what if questions, perhaps at the watershed, even at the basin scale. Another impact of wildfires is hydrologic impacts. And so definitely wildfires change the soil structure, they change the amount of runoff relative to infiltration. They also change the amount of transpiration because they remove a lot of the vegetation that would otherwise transpire water into the atmosphere. And so as a result, we tend to have more flashy systems, higher floods, more overall runoff in our system, especially under large precipitation events. And so the combination of the biogeochemistry changes in the organic matter, the increased erosion that can deliver more nutrients to the river system, and changes in the hydrologic regime can have significant dynamic changes on the overall watershed function that we would like to be able to predict. So I'd like to walk you again through our MODEX concept a little bit using this plot and have you think about how this whole idea of molecular data can actually fit into a large-scale modeling framework. And so if you think about starting up here at the top, we can use existing basin and watershed scale models, such as I showed with the Sparrow model, to provide an initial design of our network or our experimental design. As we go out into the field then and implement that design, we collect a variety of samples that are subjected to analysis, providing us with detailed omics and biogeochemical data that, as we've seen, can be put into our K-base pipeline to define biogeochemical reaction networks and kinetics. Those ultimately then feed back up into our larger scale models of the river corridor and ultimately of the watershed, providing us with updated and refined models of our large scale system, and then completing this iterative cycle of learning. Ultimately, if we have this system working well and we refine these models, we have a number of scientific opportunities before us. Can we really be able to simulate biogeochemical responses to dynamic hydrology across diverse river corridor settings, within watersheds, across basins, across continents, and even across the globe? Can we consider and account for diverse hydrologic conditions that might exist, for example, different seasons or different degrees of hydrologic variability depending on where we are in the geography? We could use this approach to answer science questions using a consistent methodology across these multiple systems. And some of the key science questions we might ask would include things like, how do these microbial communities, in our case in the hyporheic zone, respond to drying or inundation cycles, to maybe floods or other kinds of hydrologic perturbations? How do respiration rates vary across different river systems? And as we work through the WHONDRS data sets with our hands-on students this week, we're starting to gain some insights into differences between those 7 systems that have been studied. And you see here 3 of those example systems shown in the photos. I believe this one here is the Erpe River in Germany. This photo here is of the Nisqually River in Washington State, which is a glacial river near Mount Rainier. And the lower one is the Altamaha River in Georgia in the southern United States, a tidally influenced river. And so we can start to look at how the respiration rates or other features of the system might vary between these very different river systems. And how do these responses actually depend on the system type, or maybe even on the history of that system, prior hydrologic or biogeochemical history. And so hopefully that's given you a little bit of a vision of how we might use the kinds of information that we're talking about throughout the course of this study to really tackle large-scale problems. One of the things I hope you see in that is that these kinds of problems really aren't problems that we can attack as individual researchers. And so these really are critical that we approach these as communities. And so the ideas of WHONDRS, K-base, other community resources like JGI and EMSL, the whole research community coming together to work on these problems together is really critical to their solution. So because of that, we're very excited that all of you have signed up for the course and are interested in here. And we hope to keep these conversations going as we move forward on this time. So I'd like to, again, point you to the SBRSFA website. If you'd like to follow us on Twitter, we use this Twitter hashtag. And the work that we're doing on the SFA and also a number of these other resources is sponsored by the Office of Biological and Environmental Research in the Department of Energy, and in particular the Subsurface Biogeochemical Research Program. And as you can see, we heavily rely on resources like EMSL for the work that we're doing. So thank you for your attention. And at this point, we have some time for questions and discussion.
Thanks, Tim. First, you got a great presentation from Discord. I agree. Nice work. There was a question. Can you go back to the model data integration slide?
Sure.
And I'm assuming a question is brewing.
Yes, I think this is the one that would mean.
Okay. So that, Britt, you were asking for that. I'm not seeing a specific question, but maybe it's being typed. There's some questions in the, sure, yeah, you can type questions into the Zoom chat. Posting questions on Discord is preferable if possible, so that everyone can see all the questions. I can try to move questions over as well. So in either spot, just post your questions and we'll get them answered. It looks like Britt is typing. So I think a question is imminent for you, Tim.
Okay. By the way, James, you have your video on, but you're not showing.
Oh, sorry, slider. I had the slider over. Thanks. I had the slider over over the camera. I can see multiple people typing. There we go. Here you go. Is there a publication or series of publications that illustrate this MODEX cycle?
Interesting question. So I would have to think about that. Certainly, the MODEX cycle is something that's become a really it's a thread woven through the Subsurface Biogeochemical Research Program and other programs at DOE. So definitely there are papers that use this kind of approach, and certainly it's something we've used heavily in our research. I wouldn't say there's a paper that explicitly calls this out in detail. Probably the closest thing, and I can put a link to it in the Google Doc, is the one of the BER strategic planning documents, which has a detailed description of MODEX and how it's used across our program. But I don't know, James, can you think of any relevant papers we could post in that regard?
Not really. It's so funny. Like, it's such a common idea in the DOE space that, like, I think everyone just takes it for granted, which is not great. So I think we could probably dig up a few links and share them to workshop reports and things of that nature. Because like that MODEX concept and cycle, I think comes up in almost every workshop report tied to specifically to the biological and environmental research part of DOE. And so, I mean, there's the one workshop report that I can't remember, Tim, if you mentioned it, but Amy mentioned it, or I think you did mention it. Anyways, the Open Watershed Science by Design. I think there's some MODEX stuff in there, maybe referenced in there. I'll grab the link for that one at least. If I can find a couple others, I'll put them into Discord.
We do have a paper that we're working on that I think is really relevant to this, and that is our last field campaign let's see, when that was that February of this year? It was February of last year, before all of the problems happened, I guess. So basically where we used numerical models of coupled river and subsurface to predict where river exchange would be strong and in which direction, whether it would be downwelling or upwelling. And then we use that to actually guide the emplacement of a number of sensors out in the field in the Columbia River system that we're currently using to analyze the fluxes and compare with the model predictions. So I think that's one example. It's not yet published, but we're working on paper right now on that and hope to have that submitted soon.
Staying with the Modex theme, there was a question, what is the turnover rate of this iterative cycle?
Ah. Well, I think it depends on a number of things. One is like how complex is your model and your data collection. So obviously, this could happen on a very short term if you have a relatively simple model that you're tuning and fast turnaround on your experiments. I would say for our project, our experience is that this probably turns around on the time scale of months to maybe a year, because it takes quite a bit of time to go through that entire cycle of generating the complex models, predicting, using them to design experiments, collecting the data, analyzing the data, and then comparing back and updating the model. But yeah, so I would say for us that tends to occur on time scales of months to years, but it is a continually ongoing process. And in our project, we have monthly model data integration meetings where the intention is directly to address this, this MODEX activity within our project.
Cool. Let's see, here's another question. There was a slide about using neural networks for FBA and reactive transport models. Is that an ongoing work, or is there anything already available?
Yeah, that is fairly new work. I think Hyun is going to present some of that tomorrow in the metabolic modeling sessions. But that is quite a new result, and actually quite exciting result. It was something that Hyun and I had talked about when he was still here at PNNL. Going back to some old papers where we had used the output of flux balance analysis to try to generate reactive transport models using a lookup table, and with rather unsatisfying results when we think about extending that to full communities. And so we came up with this idea of, could we replace that with a neural network? And actually, Hyun's early results from this are quite striking. So I think he'll be presenting some of that tomorrow. Is that correct, Hyun? Is that included in your presentation tomorrow?
Yeah, that's right. But it's not tomorrow, but Thursday. And Thursday, I have a separate talk about it.
Okay, great.
I missed one that was kind of on the Modex theme. Do you use special tools for optimal design of experiments based on your preliminary model predictions?
So in the case of that I described, we didn't really use any special tools other than we generated the outputs of the models. We did use, in that case, we used some machine learning methods to classify hydromorphic features within the river, which we used in combination with the model results to guide our design. Certainly there are more rigorous ways, and our colleague Xingyan Chen is an expert in Bayesian information, basically data assimilation tools using Bayesian methods. And so those would be a more formal approach to use data to update models and then use potential simulations of data to guide uncertainty quantification that then could be used to tell you where you might best collect your next set of samples. So formalization of those tools, we could certainly, there are avenues we consider to do that.
Yeah, and related to that, we had an example that Xingyan was heavily involved in, of basically putting observation locations into a numerical model, and then asking, if we have observations in these places and the observations of particular kinds of data, how much information does that give us in terms of how the system is functioning? And you can take that kind of an approach to then vary your observational scheme, like where you would put sensors or where you would collect samples, and then use the Bayesian data assimilation methods to basically optimize where and when you go generate information. And so that's a nice way to couple data and models together using sort of numerical experimentation and data assimilation up front.
Mhm.
But I know Hyun has done a lot of things of this nature as well. I don't know, Hyun, if you wanted to say a couple words.
Sorry, I was looking at other slides and I couldn't.
I was just thinking about you and I've, you and others have talked many times about formal methods um, to evaluate and design experiments to make sure they give you maximal information to inform a model. Do you
Sure.
want to say a couple words on that?
Right. I recently discussed it with one of my students here that we are thinking about model-based design of experiments, and initially models are not available. Therefore, we can start as an example. Start with a linear-based statistical model to generate initial set of data that can be fed into our model, and then through iteration between experiments and model, we can better design what's the next step to generate most sensitive data that can be used for determining parameters. So we are thinking about iterative process between model and experiments.
So another sort of way of thinking about it, another way that models can be used is actually to formulate hypotheses. So for example, an example of this, James has used a model called Nexus in collaboration with Jesus Gomez Velez, our collaborator, to formulate a hypothesis about the distribution of hydrologic exchange with respect to stream order. And that information can be used then to help guide placement of sensors that we're using to look at the relative contribution of in-stream respiration to subsurface respiration. So that's another example of how a model has been used to help design an experiment.
Cool, and I think maybe we have time for maybe one more here. There's a question, when talking about these kinds of questions, this kind of science, how do you take the redox ladder in electron acceptors into consideration? Things like nitrate, iron, manganese, sulfate.
Mm-hmm. Mm-hmm. So we have been looking quite a bit at nitrate, and definitely the focus in the summer school so far has been on aerobic respiration. So with just primarily oxygen as the electron acceptor. It does turn out that when you go further down the redox ladder, one of our colleagues, Jianshu Zheng, has been looking quite a bit at how that impacts the microbial community and the response. And in particular, it seems that when you talk about aerobic respiration, there are a lot of generalists that can do that. Whereas as you go further down the redox ladder, the organisms tend to be more specialists and fit a particular niche. And so she's been doing a lot of work on trying to understand the nitrogen reaction networks from that point of view, of identifying the different specialists and their roles, and then being able to identify them through genomic analysis. Ultimately with the goal of putting that into kind of a cybernetic type of framework.
Very good. We are at 5 o'clock. There's a couple other questions I think in the Discord that we can probably answer just in Discord, I believe. So with that, I think that is the day. Tim, do you want to say any words looking forward to tomorrow? I do, before you say that, sorry. I want to thank people, of course, for joining, everyone that contributed. I felt like it was a really, I felt a really successful day in all kinds of ways. Some basics, some fundamentals, digging into some nitty gritty, and some high level concepts as well. I hope you all enjoyed it. Please keep reaching out to us with questions. The Discord channel will be open. We'll continue to monitor that tomorrow, the rest of the week. So hit questions into the metabolomics channel. We'll do our best to get back to you right away. So with that, thank you very much. And Tim, do you want to point us towards tomorrow?
Sure. So I know that it takes a lot of energy to go through all of these activities and these days. I think in many ways, tomorrow is one of the most exciting days in that we're starting to really be able to take the information that we know from metabolomics and metagenomics and put it into a model that actually predicts how the microbial community will function. And so for me, this is one of the really intriguing things that kind of got me interested in coupling microbial models with reactive transport models, and goes back some decades of study. So I really encourage people to join us tomorrow for study of metabolic modeling. Hyun is one of the world's experts in the diversity of metabolic modeling approaches, and particularly cybernetic approaches. Recently wrote a textbook on that. So we have an excellent group of researchers to draw from. Would also like to mention that our communications folks have been interested in getting your input for perhaps a video of what you've learned from the summer school. So if you're willing to answer the question in 5 seconds or less, what are you learning from the summer school, record it on your cell phone or your computer and send it to Andrea Starr. I've put the link in the Zoom webinar chat, or her email address. And so other than that, we'd love to continue the conversation over in Discord. We'll be continuing to compile links, useful links in the Google Doc for you. And definitely, if you have questions, consider keep asking, and we'd be happy to continue interacting. We'll hopefully see you tomorrow morning.